All right, Ninja Nerds, in this video, we're going to continue on with the pentose phosphate pathway and specifically focus on the regulation. Now, if you guys remember, I talked about that special enzyme. And if you guys remember, we had that glucose 6-phosphate. If you guys remember, we had glucose 6-phosphate. And he was being converted into what? 6-phosphoglucanolactone. And if you can remember, the enzyme that was catalyzing this step was going to be glucose 6-phosphate dehydrogenase, right? This enzyme was catalyzing this step here. And what's important is I told you that this enzyme is highly regulated in this step. And how we determine this is actually based upon, again, the body's demands. So here's the thing. People are wondering, okay, Zach, well, how do I know that this glucose 6-phosphate will go into the pentose phosphate pathway versus going into glycolysis? Like, what's the stimulus there? Well, glycolysis is dependent upon ATP demands, right? So glycolysis depends upon the demands of ATP. For example, if there's very little ATP, you're going to want glycolysis to increase. Whereas the opposite, if the ATP is actually too high, then what? Then you're going to want to decrease glycolysis. Simple as that, right? Same thing goes with the pentose phosphate pathway. So it goes like this instead. If you guys remember, in this step, I generate, what am I taking? I'm taking NADP positive and converting it into NADPH. Well, the whole purpose of this oxidative phase, remember there's two phases, the oxidative phase and the non-oxidative phase. In the oxidative phase, we're generating a lot of NADPHs and a lot of ribose 5-phosphate, right? But here's the significant point. You're trying to make NADPHs. So if you have low NADPHs, right? So in other words, you know when there's low NADPH, that's when the ratio of NADPH over NADP positive, who's greater in this case? Well, I have more NADP positive and less NADPH. So this ratio is thrown off. So now, whenever there's low NADPH in this condition, so we can describe the ratio concept here, is that if there is low NADPH or the ratio of low NADPH for high NADP positive, this is a stimulus. This is a stimulus to this enzyme. That will stimulate it. But the opposite can be said for whenever your NADPH is too high. So now what's going to happen with the ratio? Now I have more NADPHs as compared to my NADP positives. So in this situation, in this scenario, what's happening here? Now this is low and this is high. This situation would apply as an inhibitor. Okay, so let me actually get that stimulation out of there because that's only for this one over here, whereas this one is an inhibitor. Okay? So if you already have a lot of NADPHs, you're not going to want to keep having this pathway go through. You're going to want to inhibit this enzyme. So again, let's represent here green. Green is representing that you have high NADPH. Whereas orange is stimulatory, meaning that you have low NADPH. Okay? And these are the primary regulators of this step. Now, that's kind of general because it's not really a great explanation of how this actual pentose phosphate pathway is actually occurring. This is kind of an oversimplification. So what we need to do is we need to go through 4 different scenarios of how this actual pentose phosphate pathway is really being regulated in more detail. So there's 4 scenarios. We're going to go through each one of them step by step. The first scenario says, okay, our body's saying that we need to make ribose 5-phosphate and we don't need NADPH. Which pathways will our body take care of, okay? Okay, if you guys remember, when we talked about it in the previous video, that the oxidative phase was generating a lot of NADPH and a lot of ribose 5-phosphate. Whereas in the non-oxidative phase, we were generating a lot of ribose 5-phosphate. We didn't use NADPH in that step. That's what we want to do because we don't want NADPH. So in this step, we want to primarily depend upon the non-oxidative phase. And think about this for a second. When do we need ribose 5-phosphate? When we need to make nucleotides. All right, so whenever we need to make things that are going to be specifically for DNA, for RNA, for ATP, for NAD, for FAD, for coenzyme A, making those types of things. Okay, so let's explain how this is going to work. And we're going to go pretty quick, right? So again, if you guys remember, what can happen in the non-oxidative phase? Let's say I have glycolysis. So you remember glucose goes to glucose 6-phosphate to fructose 6-phosphate to fructose 1,6-bisphosphate to DHAP, which stands for dihydroxyacetone phosphate, which can isomerize with GA3P, but it can also get broken down. This gets converted into 1,3 BPG. This gets converted into 3-phosphoglycerate, 2-phosphoglycerate, PEP, and then eventually pyruvate. And then because we already talked about it in the other video, that out of this, you're going to generate at least a total of a net of 2 ATP from this glycolytic pathway. Now, in this situation, you guys remember that we could take glyceraldehyde 3-phosphate, right? And what could we do with that? We could combine it with fructose 6-phosphate, which I'm just showing as F6P. If you guys remember, these two can react with one another. If these two react with one another in the presence of a special enzyme, okay, and you guys will remember because it's all going to come back to you, I'm transferring a 2-carbon fragment. Which enzyme transfers 2 carbon fragments? This is the trans-ketolase enzyme. The trans-ketolase will transfer the 2 carbons from fructose 6-phosphate onto GA3P. He will then turn into xylulose 5-phosphate. What can happen with the xylulose 5-phosphate if you guys remember? He can be converted into ribose 5-phosphate. Okay, there's one. I made one ribose 5-phosphate. No NADPH. Now, I can also synthesize from this. He transfers the 2 carbons and he gets converted into a 4-carbon, which is called erythrose 4-phosphate. I'm just denoting it like this now, right? E4P. Then if you remember, erythrose 4-phosphate and the X5P reacted previously because this is a reversible reaction. Now what happens? Then we're going to take this erythrose 4-phosphate and react it with another molecule. What was that molecule that we had? Fructose 6-phosphate. These two are going to react. And they're going to react via a trans-aldolase enzyme. And they get converted. What happens? He takes his 3 carbons, transfers it onto him. He gets converted into cellular heptose 7-phosphate, and he gets converted into glyceraldehyde 3-phosphate. Then what happens? Then that cellular heptose 7-phosphate combines with the glyceraldehyde 3-phosphate in the presence of a trans-ketolase. And that transfers a 2-carbon cellular heptose 7-phosphate, transfers 2 carbons from him onto the GA3P. He gets 2 carbons. He's going to turn into xylulose 5-phosphate, and he'll turn into ribose 5-phosphate. Now what did I just do? I synthesized ribose 5-phosphate from this way. And you guys know eventually, we'll just show it here in a green, that the xylulose 5-phosphate can eventually get converted into ribose 5-phosphate. How many ribose 5-phosphates did I generate from this glycolysis pathway? I generated 3 ribose 5-phosphates from the glycolytic intermediates. So from glycolytic intermediates. Okay, that's how we would deal with this scenario. So when our body needs to make R5P, what do? You have the glycolytic intermediates feed into forming how many ribose 5-phosphates? 1, 2, and then eventually this one can get converted into the third one. So we'll just circle this X5P, which will eventually get converted into R5P. So we made 3 of them. Okay, let's go to scenario 2. The second scenario says, okay, we need ribose 5-phosphate and we need NADPH. Okay, so now This one should be easy to remember. And the reason why this should be easy to remember is because, oh, I remember from the oxidative phase, Zach was saying that we make NADPH and ribose 5-phosphate. Huh. Well, look at that. I'm going to do the oxidative phase. So I'm going to do the oxidative phase because I remember Zach saying, oh, when I did the oxidative phase, I make ribose 5-phosphate and NADPH. And let's think about this. When will we need to do this? Well, ribose 5-phosphate is needed for making, again, FAD, NAD, ATP, RNA, DNA, a lot of different things. CoA. What is NADPH needed for? Nucleotide synthesis, neurotransmitter synthesis. It's needed also for fatty acid synthesis, cholesterol synthesis. This cell could be getting to replicate, divide. We need this whenever maybe the cell's replicating. Okay, let's go through this process then. So let's say here, we now take and show you what's going to happen here. Well then, what happened with that glucose? So we have glucose here. He gets converted into glucose 6-phosphate. But the body says, oh, you know what? I'm going to take this and I'm going to shunt it into the oxidative phase. What happens in the oxidative phase? In the oxidative phase, I utilize a special enzyme, and that enzyme is called glucose 6-phosphate dehydrogenase, which does what? It converts glucose 6-phosphate into, I'm going to abbreviate it, 6-phosphoglucanolactone. Then what happens? That 6-phosphoglucanolactone gets converted into 6-phosphoglucanate. Okay, so this is going to get converted into 6-phosphoglucanate. Then what happens? The 6-phosphoglucanate gets converted into ribulose 5-phosphate. And then what happens with that ribulose 5-phosphate? You know that the ribulose 5-phosphate, I'm going to put ribulose for this one, but you know that the ribulose 5-phosphate gets converted eventually into ribose 5-phosphate through the isomerization enzyme, right? And this will get converted into ribose. I'm going to put R5P, ribose 5-phosphate. Okay, so this is the importance here. And so again, if you remember, what enzyme is here? It's not as important of a one, but it's called lactinase. What enzyme's here? It's 6-phosphoglucanate dehydrogenase. Now that's not the important points of all this. What was the important point of all this? I generate an NADPH in this step. And I generate a NADPH in this step. Okay. So what did I just do in this step, guys? I generated my NADPHs that my body needed, and I generated my R5Ps that my body needed. Okay. And why is this good again? Because the cell could be dividing. So it needs our cells to be making certain types of DNA, RNA, specific types of nucleotides, and different types of cholesterol, fatty acids, so on and so forth, okay? Okay. Now let's go into scenario number 3. So what does scenario number 3 say? Scenario number 3 says, okay, we don't need ribose 5-phosphate, so there's no need for the ribose 5-phosphate, but we do need NADPH. So if I need to do NADPH, this is a really tricky one. This one's a weird one. So we're not going to write down exactly what's happening. Actually, no, I will write down what's happening, but we're going to have to explain it in a different way. So now, 2 phases are occurring. The oxidative phase and gluconeogenesis. Okay, so 2 phases are occurring here, 2 things. Oxidative phase and gluconeogenesis. Let's explain how this is happening. This one's a little bit weird, but let's see if we can stick in there. Okay, so if you guys remember, what's the oxidative phase? Similar to this, so let's just blaze right through that. Okay, so let's just blast right through that really, really quickly. Let's do this one in red. Glucose to glucose 6-phosphate, right? Then that glucose 6-phosphate can do what? It can then get converted into 6-phosphoglucanolactone, which can get converted into 6-phosphoglucanate, which can get converted into, I'm going to put RI5P, which simulates that that's ribulose 5-phosphate, and then that gets converted into ribose 5-phosphate. Now, in a different color here, I'm going to remember that your G6P, he eventually gets converted into fructose 6-phosphate, and then into fructose 1,6-bisphosphate, then to DHAP, then into GA3P, which is your glyceraldehyde 3-phosphate, and then eventually to pyruvate, right? Okay. In this step, which ones? Specifically this step here, when I go from glucose 6-phosphate to 6-phosphogluconolactone, I take NADP positive and I convert it into NADPH. Okay, cool. And then from 6-phosphogluconolactone to ribulose 5-phosphate, I take NADP positive and I convert it into NADPH. Cool. And if you remember, just to reiterate, this is glucose 6-phosphate dehydrogenase. This is lactonase. And this is specifically 6-phosphogluconolactone dehydrogenase. Now, I made R5P. Well, I just said I don't want to make R5P. What do I do with that? Watch this. You guys remember all those steps that we covered? That eventually, we're not going to go through every single one of them, but you guys realize that all that non-oxidative phase is reversible. So eventually, what can I do with that R5P? Let's do this in a nice pink color. I can eventually take that ribose 5-phosphate and I can convert it into glyceraldehyde 3-phosphate eventually, right? And in the same way, I can take that ribose 5-phosphate and also convert it into what? Fructose 6-phosphate. Now what can happen? This step is reversible, so I can take glyceraldehyde 3-phosphate into fructose 1,6-bisphosphate. Then if I want to go from fructose 1,6-bisphosphate to fructose 6-phosphate, it is regulated by the fructose 1,6-bisphosphatase enzyme, but I can go backwards, and then I can go backwards here, and depending upon if we are in the liver, the kidneys, or the small intestine, there is a special enzyme here that can convert the glucose 6-phosphate into glucose, and that's called the glucose 6-phosphatase. But eventually what happens is R5P can make GA3P and fructose 6-phosphate, which can eventually be converted into glucose. And what happens is I use up all my ribose 5-phosphate, so I don't have any of him. So what's the whole purpose here? All I really did is generate a lot of NADPHs and used up my ribose 5-phosphates to make glucose. So it's an intelligent design our body has. And this can occur based upon our needs. Maybe we need to have a lot of biosynthesis reactions because we need to make NADPH for synthesizing fats or synthesizing cholesterol or synthesizing neurotransmitters or synthesizing nucleotides, you get the point. Or our body needs glucose because I don't need any more R5P. Okay, now last scenario number 4. Let's say that our body needs NADPH and it needs ATP. It's going to be very, very similar to this one. Let's do this one in green. So now, same thing, glucose to glucose 6-phosphate to fructose 6-phosphate to fructose 1,6-bisphosphate to DHAP, glyceraldehyde 3-phosphate, and again, these are isomerized. And then eventually you realize that this can go to pyruvate. And you know that out of this, I generate what? A net of 2 ATP. Okay. Same thing like we've talked about. Glucose 6-phosphate can get converted into what? Let's actually do this in a different color since we've used this one already. Let's do this one in blue. So glucose 6-phosphate can be acted on by glucose 6-phosphate dehydrogenase, which will do what? Take NADP positive and convert it into NADPH. Okay, cool. And I make 6-phosphogluconolactone. Then I can take that 6-phosphogluconolactone, convert it into 6-phosphogluconate, and then I can convert that into ribulose 5-phosphate, and then I can convert that into ribose 5-phosphate, just like I did in the previous one over here. But now, watch this. Oh, and then again, what is this enzyme here called? This one is called 6-phosphogluconate dehydrogenase. Why is that important? Because I generate a NADPH from here. So let's kind of, bubble these guys up here because I made my NADPH. So I took care of that part of my body's demands. Now, what's the problem? Okay. Now what I'm going to do is I'm going to take this ribose 5-phosphate. And depending upon the body's needs, instead of them making glucose, I can take that ribose 5-phosphate, convert it into glyceraldehyde 3-phosphate. What else can I do? I can convert it into fructose 6-phosphate. Where can these guys go? they can go down. They can go from fructose 6-phosphate into fructose 1,6-bisphosphate. That can go down into DHAP or diglyceraldehyde 3-phosphate, and that eventually could go down into pyruvate. And what will that do? That'll help to generate ATP. And then we take care of the body's demands because it needed ATP. So this is the way that the body can do this. So again, now we know all of these scenarios, okay? And then what would be the situation for this? We would want to do the oxidative phase here. And what else would we want to do? Glycolysis. Okay. Last thing I want to talk about, guys. Why is all this important? Because just knowing all these things, okay, that's cool. It's a cool thing to know. But why is this significant? Why is this important? Okay. It is important, again, because if you guys remember, just real quickly, NADPH is important for what things? He's important for a lot of different pathways. Okay, what is it? Lipid synthesis, cholesterol synthesis, neurotransmitter synthesis, and even nucleotide synthesis, right? So a lot of different things he's used for. Okay, what is significant about the R5P one more time? Okay, so now for R5P, ribose 5-phosphate, he's significant because if you remember, he can do a boatload of stuff, right? He can help with DNA synthesis, RNA synthesis, ATP synthesis, NAD positive and FAD synthesis, and also coenzyme A synthesis. So he's involved in all these reactions. So you can see how going through all of these scenarios, you can basically determine what the body will do, which one of these actual pathways will occur based upon the body's demands for these two molecules, or for ATP needs also, as we saw in scenario 4. Okay, last thing here. Our body's very, very interesting. We have a way of controlling something. What did I tell you was one of the really cool ones I told you that we were going to mention? Free radical reactions. Why is this important? Because free radicals are extremely dangerous. Let's go through a quick free radical pathway, extremely quick. Oxygen, you know, whenever you can actually have it at the electron transport chain certain times, he can actually be converted into what's called a superoxide anion, which just means he got an electron plucked out. So now he's a radical. Now, when this happens, two things can happen. One thing is I can convert this into a very deadly chemical called peroxynitrite. But this will only happen if I have the presence of a nitric oxide radical. So you know, nitric oxide radical can also be formed within our body. So here's our nitric oxide radical. This nitric oxide radical can combine with this molecule. So what is this called? This is called superoxide anion, which is a radical. This is our nitric oxide radical. When these combine, they can make peroxynitrite. Why peroxynitrite? Why is that dangerous? Because this can cause very, very dangerous effects. It can oxidize proteins, which can frick them up, destroy them. It can nitrate proteins, which can alter their activity. It can inactivate enzymes, maybe that control like channels within the body. And the dangerous part, it can cause lipid peroxidation, which can cause very, very dangerous effects also to our cell membranes. Okay, but our body's very interesting. It has a way to deal with this superoxide anion. What it can do is it has an enzyme here. This enzyme is called superoxide dismutase. So it's called superoxide dismutase. What superoxide dismutase also consists of manganese within it. It's important. It takes the superoxide anion and converts it into a less dangerous molecule. Less dangerous molecule. Still dangerous, but not as dangerous. This is called hydrogen peroxide. So again, what is this molecule called? Hydrogen peroxide. Now, there's 3 destinations of this guy, okay? One is this hydrogen peroxide actually be converted into another dangerous molecule. This extremely, probably one of the worst ones, is called a hydroxyl radical. This is called a hydroxyl radical. the hydroxyl radical can cause DNA damage. So those free radicals that you generate from the actual hydroxide free radical can damage DNA, it can damage the lipids, it can cause lipid peroxidation, and it can also damage our proteins and enzymes. And this can lead to a lot of cytotoxic effects, killing the cell. Now, this can happen, we can actually control this via the Fenton reactions. So Fenton reactions can occur in certain steps over here. Now we'll just put that. So Fenton, or you can put some iron in there, Fenton reactions. Anyway, we're not going to talk too much about that. Here's the important part. The hydrogen peroxide can have another thing. You know, there's enzymes called catalases. We talked about this in the peroxisomes. The catalases can convert the hydrogen peroxide into, water and oxygen. But here's where and then technically it's 1/2 of an oxygen if we were to be really, really particular. But here's where it's important. This step here, taking this hydrogen peroxide and converting it into another molecule. Okay, I can also convert it. Let me actually do this in a different way. I'll do it like this. I can convert it into water and oxygen also, but another special enzyme. This enzyme is called Glutathione Peroxidase. And this enzyme is actually helping to convert hydrogen peroxide into water and oxygen. Now, here's the problem. Here's what can happen. We need to talk about another enzyme that's extremely, extremely important. Last enzyme. Because the default in this enzyme can cause disastrous effects on the body. So let's do it over here. Okay. Let's get this out of the way. Alrighty. So now there is a molecule in our body called glutathione. Now glutathione, I'm just going to draw it as I'm going to put a G. So I'm going to represent this. You're going to see it from now on as a G. You're just going to see it as a G. Glutathione is special because if I have a glutathione, technically in its reduced form, it has this thiol group. So this is its reduced form. Glutathione is our natural antioxidant in the body. But you know what happens? You know those hydrogen peroxides and superoxide ions? So you remember the hydrogen peroxide that you had here? This glutathione can react with this hydrogen peroxide. And when it does, it actually can convert this hydrogen peroxide into water and that oxygen. It can also do this with other different molecules. But there's another enzyme involved in this step. This enzyme is called glutathione. This is called the peroxidase enzyme, right? So we talked about that one over there. But when this glutathione in the reduced form unloads its hydrides, what's hydrides again? It's basically a proton and 2 electrons. And it unloads it onto this hydrogen peroxide. And it helps to convert it into water and 1/2 of an oxygen. What happens to him in that same process is, look what happens. He gets converted from the reduced form into a different form. Look what happens to him. I take a glutathione molecule and another glutathione molecule, and now I'm going to link them together, okay, through this disulfide bond. So look what happened here. Glutathione, I might have 2 of these. Let's say that I have 2 individual one of these. And what are they doing? They're taking and dropping off hydride ions onto this hydrogen peroxide or other free radicals to convert them into less toxic forms. But in the process, they get converted into the oxidized form. Why is that bad? Because then they can't drop any hydrides onto the actual what? They can't drop any hydride ions onto this hydrogen peroxide. So now it's not going to be able to get rid of these free radicals. This is where these NADPHs come in. So now what I'm going to do is, I'm going to do something cool. Let me actually show, because I'm going to show it here. I'm going to convert this guy back to this guy. But let me show you exactly how that's happening right up above this. So look at this. I'm going to have glutathione with a thiol, glutathione with a thiol group. Okay, that's how we're going to represent it. Then it's going to react with the hydrogen peroxide to convert it into what? H2O and 1/2 of an oxygen. But then what happens? This gets converted into the oxidized form of glutathione. In order for me to go back to this, I need those NADPHs. They're going to do what? They're going to drop those hydrides off onto the glutathione in the oxidized form. Now what's going to happen? It's going to go back to the reduced form. That's amazing. That's the way our body deals with these free radicals. Those NADPHs that we're making from the oxidative phase, that we're making from the Pentose Phosphate Pathway, right? We're going to use that to drop onto these oxidized glutathiones to convert them back into the reduced form. And the enzyme involved in this step is called Glutathione Reductase. Called Glutathione Reductase, which should make sense because it's reducing the glutathione. Now, why is this bad? If you guys remember in anemias, we talked about this. Whenever there is a deficiency in that enzyme, where's that enzyme at? Let's find him. This guy. When there's a deficiency in this enzyme, you're not making this enzyme, glucose-6-phosphate dehydrogenase. Due to some type of mutation in which this enzyme is lost, can you make NADPHs? No. If you can't make NADPHs, can you unload those onto the glutathiones involved in the free radical reactions? No. What happens? You lead to the formation of Heinz Bodies. Remember Heinz Bodies? We talked about it in anemias. If you guys watched that video, it's basically you're going to have those reactive oxygen species. So remember you have all those reactive oxygen species, like the superoxide anion, the hydroxyl radical, the peroxynitrite, so I'll put peroxynitrite, tons of different, even what, hydrogen peroxide even accumulating. It's basically damaging what molecule inside of our red blood cells? Hemoglobin. And what was it doing to the hemoglobin? It was causing that hemoglobin to stick to the wall. Remember we had a red blood cell here? Let's say here's my red blood cell. Remember it was actually causing the actual hemoglobin to get stuck with the inner wall of the actual red blood cell membrane, making it less pliable, less flexible. What did that result in? The overall result was it led to hemolytic anemia. And then hemolytic anemia is very, very dangerous, right? Because it can cause a lot of problems, low oxygen carrying capacity. It could actually cause the hemoglobin to be released into the bloodstream and can actually also cause the iron to get released into the blood, which is also very toxic. So there is certain conditions in which people actually eat fava beans. There's some chemical in the fava beans that actually reduce the G6-PDH and other different types of things. But the whole purpose is that the pentose phosphate pathway is a beautiful pathway and our body really does depend upon it in order for us to be able to do a lot of different activities. All right, engineers, I hope you guys enjoyed this video. I hope you guys I hope it made sense. If it did, guys, please hit that like button. Please subscribe. We really depend upon you guys subscribing because we look forward to doing this. Also, comment. We look forward to hearing from you guys. All right, engineers, until next time.